Aging, and Age-Related Disease. He gave the first talk at the NIH on a reversal of human aging, published the first article on the potential of telomeres as a clinical intervention, and authored the only medical textbook in this field, Cells, Aging, and Human Diseases, by Oxford University Press. He was the editor-in-chief of Rejuvenation Research and the director of the American Aging Association, and as well as the editor-chief OBM Geriatrics. He has authored more than 100 books, chapters, and articles, including The Telomeres Revolution, which the Wall Street Journal praised as one of the best science books of the year. He is the president of Telocite, a biotech firm taking telomere therapy of FDA human trials. As well as author of A Unified Model of Dementias and Age-Related Neurodegeneration, published in Alzheimer's and Dementia, the Journal of Alzheimer's Association in January of 2020, which generated more than 600 reprint requests in the first 2 weeks. And his title will be Age-Related Disease, Moving Towards a Cure. Thank you.
Thank you very much for having me, Krishna. Let me go ahead and share my slides, if I might, with you.
Thank you, Dr. Michael.
You're all set. Can you hear me all right, Krishna?
Yeah, we can hear you.
Very good. Yeah, I sort of enjoyed that last talk talking about COVID, because I'm probably the only person you know who actually has been immunized against COVID more than a month ago. I volunteered for a trial, and I was delighted that if I got the placebo, I certainly got fevers, chills, headache, and fatigue. So I don't think it was saline. I think I was immunized. So I'm delighted to hear that. But let me move on and tell you about where we're going in this talk. There are basically 4 areas we're going to talk about. What I call assumptions of understanding, where we often think we understand a disease, for example, but we're not looking deeply enough. We're going to talk about unified systems models, what I mean by that, and why they're much more effective than simply going after components. Understanding the aging process itself at a very fundamental level. And finally, optimal points of intervention in age-related diseases. And in a general way, I'll be talking about aging and age-related diseases, but specifically, I'm going to focus in on dementias and much more specifically on Alzheimer's disease. I got intrigued by this entire area almost 50 years ago when I was at Stanford, and I was teaching in the medical school, and I found that although I was starting to work on the development of the nervous system, which is fascinating, but I found that when I talked about the other end of the spectrum, that is age-related diseases and aging in general, people invariably would shrug and say, well, it just happens. And I find that when people have that sort of blasé attitude, it usually means they're not paying attention to the complexity involved and that there's much more to it than they think it is. There is. So in looking over at lifespan and our effect on lifespan, let me point out a couple of things. One is there's been an enormous impact on the mean lifespan of human beings over the past, say, 200 years, going up from about a quarter century to a half century to about 3/4 of a century now, but no impact whatsoever on the estimated maximum lifespan of human beings, which is about 120 years. But more importantly, I'd like to point out that what is important here is not the quantity of life, that is the length of the lifespan, but the quality of life. And if I look back historically and ask what has had the biggest impact on the quality of life for human beings, it is not technical advances. It are not things like robotic surgery, heart transplants, the latest statin drug. Those have had an impact on quality. But the biggest impact has not been technical advances, but conceptual revolutions. Specifically, for example, microbial theory, which allowed us to have things like hand washing, which drastically cut both the fetal and the maternal death rates. The earliest sterile surgeries, the earliest rabies vaccines, the earliest antibiotics, and so on. Not only have these had a huge impact on the quality of life, but as opposed to recent technical advances, they've actually lowered the cost of medical care, not raised it. Example, in 1950, there were estimates that in a number of countries, polio would bankrupt the healthcare budgets of a number of countries because by the year 2000, with the cost of iron lungs and rehabilitation care and braces, nursing care, pulmonary care, we wouldn't be able to afford it. That did not happen. Current estimates are that per capita costs of treating polio worldwide are about 10 cents a head. I think that the same thing is about to happen when we look ahead with what's going on with age-related diseases. Currently, in many developed nations, the estimated cost of treating an Alzheimer's patient in the last year of life and not being able to affect the course of the disease is in excess of $100,000 a year, which is completely unaffordable. And a number of people have come to me about that to give statements about this in the UK, India, and elsewhere. It is not affordable. On the other hand, I don't think it will happen. Our best estimate is that we'll be able to drastically undercut the cost of medical care for age-related diseases globally, nationally, and personally, but only if we actually understand the aging process itself and be able to lower those costs. This is a friend of mine that some of you will know. This is Dr. Leonard Hayflick. He was the first person to describe cell aging in about 1960. And he and I were having a chat a year ago about this, and he made this statement. And he is absolutely right. Almost all of us, I think, without exception, would say that aging itself is the best predictive risk factor for age-related diseases. But invariably, then, we ignore the aging process and focus on histology, obvious pathology, small molecular drugs, rather than delve deeper into the aging process itself. Here's an example of an assumption of understanding. In 1980, when I started my medical career, the assumption was that almost all gastric and pyloric ulcers were caused by acid and stress. Now, it was not that we were stupid or superstitious or ignorant. We simply made the assumption that we understood the disease. And it wasn't until the British Medical Journal published this article by Robin Warren and Barry Marshall in 1983, for which they deservedly won a Nobel Prize, that we began to understand that these diseases were actually the outcome of an infection, in this case, Helicobacter pylori, seen here at the right. But again, I want to stress that we do this again and again in medicine and science. We assume we understand something without looking carefully at our assumptions. Here's a scientific example. This is a statement made by Lord Kelvin, probably in 1895, one of the world's leading physicists. And he was not ignorant, superstitious, or as I say, stupid. Very bright man. He understood weight, he understood air pressure, he had a limited understanding of aerodynamics, and was proven decidedly wrong about 8 years later, although it took about another 5 years before the world agreed to that. But again, I want to stress that he had a reasonable assumption, but he didn't examine it very carefully. So what we need to look at more carefully is underlying causes. Now, let me give you a kind of an example in medicine. This is plague. Now, plague clinically has at least 3 syndromes, and they tend to overlap a bit, but typically there's bubonic plague, pneumonic plague, and septicemic plague. And if you knew nothing about microbial disease, you would assume there were 3 different diseases, and they're not. They're all caused by Yersinia pestis. The same thing is probably going on when we look at age-related, for example, neurodegenerative diseases. So these are typical syndromes of age-related dementias. We look at Alzheimer's, Parkinson's, frontotemporal, vascular dementias, and we'll come back to some others as well. But we have begun to realize very clearly over the past century that patients with Alzheimer's disease frequently have motor deficits. Patients with Parkinson's frequently have cognitive deficits. Frontotemporals tend to overlap. All of these patients at autopsy will often show vascular changes. So it's become clear that when we look at the dementias, while there are these clinical syndromes, there tends to be a degree of overlap. And in fact, if we look at things like Lewy body and posterior cerebral atrophy and so on, we find the same sort of thing. And the result has been that in the last decade or so, we've begun to get a global consensus that there are not only atypical and mixed dementias, that is, some people don't fit in any of the categories, some people have mixed components of all of these, and there are professional interest groups and in fact global conferences on atypical and mixed dementias, but that something else is going on. And that in the same sense I alluded to before about plague, there are probably some underlying commonalities of function going on in these diseases. So what we're looking at is age-related neurodegenerative disease. And the question is, at some much more fundamental level, what is going on that results in all these age-related diseases? Well, about 1.5 years ago, the Alzheimer's Association invited me to help organize a conference. And the conference was on theoretically animal models of dementias, in this case, Alzheimer's. And the classic statement is this: everything works in mice, but nothing works in humans. And in fact, anybody involved in the research realizes that not everything works in mice either. The question, though, is why? Why is it that we get successful results in animal studies, but nothing coming out the other end in translational studies for human beings, nothing that's effective? And I pointed out a number of reasons for this. For one thing, we only share about 75% of the protein expressing genes with mice. The other 25% we don't. And many of the models that we use rely on mechanisms, physiologic mechanisms common to that non-shared 25%. But the result was that I also talked about how we needed a model, a unified systems model to explain how these diseases work, so we actually understood what was going on. The result of that was I was invited to write this paper. It turned out the person who invited me to write this was the editor-in-chief of Alzheimer's and Dementia. He promptly recused himself from the editorial process and asked to join our scientific advisory board, which he did. The paper went through 8 different editors, all of whom liked it, which I'm not used to. And in fact, they wanted me to expand the paper a fair bit, which I did somewhat. And as Krishna already pointed out, the result of this was more than 600 reprint requests in the first 2 weeks and a lot of invitations to give talks like this one. This is the sort of thing I was pointing out. I was pointing out that if you look at mice versus humans, for example, or rhesus or a number of other animals, what you find is that they have different intermediate pathways. They share certain fundamental mechanisms in the way those cells work. But if you look at the pathways, for example, the way that mice process beta amyloid is different than ours. So small wonder that if you study beta amyloid, you don't get the same results from mice you get from humans. And this is true no matter what you look at almost, unless you're looking at fundamental mechanisms. The same thing is true if we look at humans. Sorry, that took me right to the end. My fault, too early in my morning for you guys. Same thing, though, is true in humans. If we look at humans, what you find is that you can see vascular dementias, Alzheimer's, Parkinson's, frontotemporals. And what we're seeing is that all humans have slightly different genetic and epigenetic backgrounds. So they have different clinical presentations, but they still share some fundamental mechanisms that are probably responsible for what we're looking at clinically. Well, when I talk about a unified systems model, there are 2 main features it has. One, it has to be unified. It has to be able to explain everything. And the other is it has to be a systems model. Now, when I say a unified model, a model that's sufficient to really explain Alzheimer's has to be also able to explain the heterogeneity within dementias. For example, we know patients who get Alzheimer's disease at age 45. Most people say in their 70s. Some people don't get it at age 100. And the question is, why is there this heterogeneity in the presentation of Alzheimer's, or for that matter, Parkinson's or any other dementia? The same thing is true of the heterogeneity between dementias. Why is it that 2 apparently identical patients, one will get Alzheimer's, one Parkinson's, and one will get nothing? Why is that there's heterogeneity between the diagnoses? Also between species. If we look at almost any species, particularly mammal species, we find cognitive decline that's age-related. Why is it that it's expressed in different ways? Why do mice not get the same beta amyloid presentations that we get? Also, multiple upstream mechanisms. Why is it that head injury increases your risk, but we don't know why? Why is it that infections, for example, encephalitides, as alluded to in the last lecture, increase your risk, but not always? Why is it exposure to toxins, for example, paraquat, increases your risk for Parkinson's? Why does this happen and yet not always? Why do these mechanisms express themselves in disease? Also downstream biomarkers. We know that most Alzheimer's patients demonstrate tau tangles. Some demonstrate less in the way of beta amyloid plaques than others. Why is there this difference in the way these are expressed downstream, and why do the downstream biomarkers occur? It's well and good to say that we get beta amyloid plaques, but why do we get them? It's fine to say we get tau tangles, but what is the exact mechanism, and can we explain it? Furthermore, we need a model that's a systems model, not just a component model. And to give you an example of this, this is an engineering example. This is an Airbus 380 cockpit, and in the upper right, you're looking at a Rolls-Royce Pearl 15 jet engine. In the lower right, you're looking at the fan blade in the foreground from a Trent 900 turbofan engine. Now, if that fan blade fails, and I work with someone who is responsible for the overall control of this design, if that fan blade fails, we should not be asking what's just wrong with the fan blade, but what's wrong with the fan blade in the system. That is, what's wrong with that fan blade in the Trent 900, in the Rolls-Royce Pearl 15 jet engine, in the Airbus 380, at 35,000 feet, going hundreds of miles an hour at a high RPM and a high temperature. In short, it's a systems issue. The same thing is true biologically. Too often we focus in on components, histological components or pathologic components, without asking ourselves what's going on in the entire system. Here's an example from medicine. This is the Ebola River, and as you might guess, these are symptoms of Ebola. I taught a semester-long course on it. But all of these are signs and symptoms of Ebola, but they are not the disease itself. The disease itself is a very complicated process, an interaction between the virus and a complicated mammalian organism, namely you and I. The same thing is true if we look at COVID. These are common signs and symptoms of COVID. But These are not COVID. They're simply biomarkers, signs and symptoms, outcomes, components, if you will. They are not the process. The process itself is different. So for example, you can't simply cure COVID by treating fevers, chills, cough, shortness of breath, fatigue, body aches, and so on. You actually need to treat the underlying disease process. Here's another example moving to aging. These are commonly known biomarkers of aging at a macroscopic level. We see them in our cells, we see them in our patients, we see them in the grocery store. Declining glomerular filtration rate, osteoarthritis, osteoporosis, gray hair. They're not aging, but they are biomarkers of aging, but that's not the same as the process itself. If we look at the microscopic level, you see the same sort of thing. Declining rates of production of ATP versus reactive oxygen species, acetylation changes, telomere shortening, declining DNA repair rates. These are not aging. They are biomarkers of aging. We can measure these, but they're not the process itself. It's the process itself we need to understand. So biomarkers are not aging. These are common 9 biomarkers listed by a company I won't mention, but they're stressed in some of their meetings, they're stressed in the literature and their website, but they are not aging. They're simply biomarkers of aging. They do not tell you what's going on at a fundamental level. In the dementias, these are common biomarkers for human dementias. Beta amyloid plaque, alpha-synuclein, failure of the blood-brain barrier, lymphatic changes, mitochondrial dysfunction, but these are not dementia by themselves. Even Alois Alzheimer's, some more than 110 years ago, warned us against taking beta amyloid plaque as the cause of the disease. It is a biomarker of the disease. It's not the disease itself. The same is true if we look at the genes. These are genes that have been commonly linked to Alzheimer's and other dementias, but they are not the disease. For example, you can have 2 ApoE4 alleles and still not get Alzheimer's, even though you have a higher risk. You can have an ApoE2 and an ApoE3 allele and still get Alzheimer's, although the odds are extremely low. So these are biomarkers. They're common biomarkers, components, outcomes, mechanisms, but we don't know what they are, and they're not the disease itself. They're simply associated findings. Let me stress this overall. In science, we often say that if you can't measure it, it's not science. And that's true, but it doesn't go far enough. In science, it is not enough to measure it. You need to understand the process behind it. In the 1500s, Vatican astronomers measured planetary motions exactly, but they still had no idea that the Earth rotated around the sun. So the same is true in science generally and in medicine specifically. We cannot understand Alzheimer's, we can't treat it, but if we do understand it, then we have the potential to intervene effectively. So the question then goes back to the one I originally asked, which is, do we truly understand aging? Because if we do, there's a potential for being able to intervene effectively in age-related diseases. I gave the first talk ever on the aging process in 1996, 24 years ago, at the NIH in Bethesda, Maryland. And what I stressed was, again, that we do not have a fundamental understanding of the aging process. We tend to assume that it just happens, it's entropy, you get old, things fall apart, what can you expect? Which is insufficient. So that resulted in the first book on this, the first papers in Journal of American Medical Association, the textbook that was mentioned earlier. I recommend you not read it, it's pretty academic. And the book that came out a few years ago that's meant for the general public. It's something I've been intrigued by for a long time. And the fundamental question is, again, what is aging? These are aging species. Humans in the upper right, typical lifespan of about 75 years for an average. That is Coco the gorilla, a typical lifespan a little less than half a century. That Coco was actually a gorilla I babysat for a year. She spoke sign language. I am mortified to admit she had a bigger sign language vocabulary than I did. She died about a year or two ago. I will miss her. Common domestic animals, dogs and cats, for example, tend to live for about 2 decades, depending on the breed. Common laboratory rodents, about 2 or 3 years, again, depending on the particular breed. So if you wanted to say that entropy causes aging, you'd have to explain why entropy works at different rates in different species. Now, most people at this point will shrug and say, well, it's genetic. What can you expect? Which is not an answer. Genetics play a role in how we age at different rates, but that's not an explanation. That's simply one step down. Nor is this difference in aging rate confined to between species. It also occurs within species. Example, these are progeric children. This is a group of 30 progeric children, and I took them for a tour of the White House here about 2 decades ago. I used to gather them together from all over the world. Their typical age at death is 12.7 years. They die overwhelmingly of age-related diseases. The children you're seeing there are all bald. The ones with wigs on don't actually have hair, except for the one woman who actually doesn't have progeria there. They have thin skin, prominent veins. They have osteoarthritis, osteoporosis. There's a child in the upper right corner who died later this same year of a massive heart attack at age 7 while throwing a ball with his father in the backyard. Why do these children get early aging? Well, we know the gene. It's a lamin A gene. But notice that that's not an explanation. It's just a label. It doesn't tell us how the lamin A gene works and how it results in these sort of clinical outcomes. So the question still is, what is aging? Furthermore, let me point out that aging is avoidable. There are systems that don't age. Every single one of you who's listening to me are the result of a fertilized ovum, and the ovum derived from your mother. It was at that point a few decades old. It derived from her mother and her mother on back almost infinitely. In fact, back 4.5 billion years. So the line of cells that you represent is 4.5 billion years and did not age in any way for 4.5 billion years. And yet there was an inflection point a couple of decades back for each of you, and you began aging. The same is true of your mitochondria. Every mitochondria in your body right now is 1.7 billion years old. And those mitochondria never aged for 1.7 billion years. And yet were I to measure cell function and mitochondrial function in your body right now, and I'll come back to mitochondrial function, I would find that there are age-related changes. Why is it that aging did not occur for billions of years and then does? So what is aging? It is generally a balance between entropy and maintenance. It's not simply entropy, it's a matter of keeping up with it. In fact, this is an analogy from my gardens. These are gardens of mine. If I have a good garden, it is not simply a matter of not having weeds. It's a matter of weeding. That is, it is weeds versus maintenance. Maintenance is critical. And the same is true in a biological sense. To quote Lewis Carroll in The Red Queen, it takes running full time to stay in one place. In fact, the same could be said with regard to biology in a general sense with the Red Queen theory, which is it takes all the maintenance you can do simply to keep up with entropy. But that's precisely what goes on in biological systems. For example, germ cells. The germ cell line flawlessly maintains itself in the face of entropy. Whereas if I look at the 80-year-old hand on the left in this screen, what you see is a whole series of cells. And whether I'm looking at the keratinocytes, the fibroblasts, the vascular endothelial cells, the macrophages, the T cells, you name the cell, you're seeing age-related changes because maintenance has not kept up with entropy. And the question again is, why? What's going on at a deeper level? Well, if we look at that deeper level, what you see is that young cells have long telomeres and they have a specific pattern of gene expression. Now, the difference between my cells at age 7 and my cells now, I turned 70 last week. The difference between my cells at age 7 and my cells now at age 70 is not different genes. It is that the telomeres are shortened in many of the somatic cells and the pattern of gene expression has altered. And the result is a dysfunction in my maintenance. Now, you could look at this as a symphony orchestra. The difference between a young cell and an old cell is not different genes, nor is it different instruments in the orchestra or a different first violinist or oboe player or pianist. The difference is the score that they're playing, and more specifically, the tempo. In some respects, the telomere is the conductor for the orchestra. It is not the genes, but it's the conductor for the orchestra. It sets the score and the tempo. And the tempo turns out to be a good analogy because no matter what molecular pool you look at your body, whether it's beta amyloid and microglia and neurons, or whether it's collagen and elastin in the cells, between the cells and skin of my face, no matter, or beta or myeloid, no matter what molecule I look at, what I'm going to find is that turnover in young cells is very rapid, but turnover in older cells begins to slow down. So in young cells, you are continually recycling molecules. And the result is you don't get much in the way of damage. But as the recycling rate slows in molecular pools with age in cells, you find that you get gradually an increase in the amount of damage. And the result is dysfunctional molecules, for example, proteins, lipids, and dysfunctional cells. And in very old cells with shorter telomeres, altered pattern of gene expression, slower turnover, you get much more in the way of damage accumulation and dysfunction. In fact, you can look at it mathematically. This is taken from my textbook. What it really says is that young cells have a rapid rate of turnover, very little damage at equilibrium. Older cells have a slower rate of turnover and a much higher accrual of damage at equilibrium. So what you're seeing in young cells is very rapid turnover. And as the cells get older, you see slower and slower turnover. And the result is an accumulation of damaged molecules. And the same thing is true of age-related diseases. The curves intersect very nicely. Now, let's turn to a specific organ, which is the brain. In fact, we'll look at Alzheimer's. So if we're looking at Alzheimer's, one of the things we can look at is beta amyloid. We can look equally at tau tangles, and we will allude to it a little later. But if we're looking at, for example, beta amyloid, you find that in young cells, the rate at which your cells, for example, microglia, produce, secrete, rebind, reuptake, and degrade beta amyloid molecules is very rapid. But as our cells get older and as gene expression changes, you begin to get a slower rate of all 5 of these processes, and they've been measured. The result of that is you begin to see this gradual accumulation of beta amyloid microaggregates because the molecules are a little sticky and some of them sit around a little too long. You might ask, what effect do genes have on this? For example, if I'm lucky and have 2 ApoE2 alleles, what I find is that I don't make very sticky molecules. So the rate of accrual of damage as my cells get older is relatively small. But if I have 2 ApoE4 alleles, I tend to make stickier molecules. And the result is, as my cells begin to slow down their rate of recycling as I get older, I'm apt to have more and more in the way of microaggregates. And the result of that, as we all get older, is this, beta amyloid plaques. And the result of that is synaptic loss, dysfunctional neurons, dead neurons, and ultimately the clinical dementias, for example, Alzheimer's. And the same is true not only of beta amyloid, but if I look at tau tangles, alpha-synuclein, mitochondrial dysfunction, inflammatory changes, microglial activation, you name the biomarker, we see the same thing occurring and for much the same reason. Speaking of mitochondrial dysfunction, for example, reactive oxygen species, the question is, what does this have to do with aging? Is this a fundamental process? And what you find is that 99% of the aerobic enzymes that are responsible for mitochondrial energy all derive from the nucleus, and they all show a declining rate of recycling. So the point is, they are in a very dangerous environment in the mitochondria. They begin to get dysfunctional and they're not recycled as quickly. So the result is you get more and more dysfunctional anaerobic or aerobic enzymes. And the result of that is less in the way of ATP and more in the way of reactive oxygen species, that is free radical production, nor is that all. Turns out the lipid molecules that protect the cell from the mitochondria are also becoming leakier because those molecules are turned over more slowly. And the scavenging molecules responsible for capturing reactive oxygen species, for example, catalase, superoxide dismutase, are also being turned over more slowly. So is there a fundamental process going on here? You make more in the way of free radicals, you have leakier membranes, slower scavenging, and as a result, the other damaged molecules are turned over more slowly too, all as a result of age-related changes fundamental to the nucleus. Now, almost all the molecules in your body, you simply recycle. You don't repair, except for one, and that is DNA. In the time since you and I have been talking over the last 27 minutes, every single cell in your body has had some DNA damage, which you have flawlessly repaired. You have 4 sets of enzymes, families of enzymes responsible for that, for detection, excision, replacement, and ligation of the damaged DNA base. Now, all 4 of those families also show slowing of recycling as you get older. So you have a slower detection multiplied times slower excision times slower replacement multiplied times slower ligation. And the result is an exponential increase in the rate of DNA damage and therefore malignancy. And in fact, it's not a matter of years. You see this exponential increase in malignancy in mice over 2 years and humans over 100 years. The question then is not how long you live or what's your lifetime exposure to carcinogens. The question is, how slow is your DNA becoming over time? So cancer with age goes up exponentially, but largely as a result of decrease of DNA repair rather than simply exposure to carcinogens. Now, I've alluded to telomeres. Let me make 2 statements bluntly. One is telomeres don't cause aging. Telomeres no more cause aging than genes cause evolution. Genes don't cause evolution, but they're a key player in evolution. Evolution is a remarkably complicated process that goes well beyond simply identifying genes. The same thing is true of aging. Telomeres play a central role in the aging process, but they're not the cause of aging. To use the verb cause here is a bit naive. It underrepresents the complexity of the process going on. Furthermore, telomere length is irrelevant. It is not telomere length that matters, it's the relative length or the change in length or the rate of telomere loss. So for example, there are varieties of mice that have telomeres that at birth are 10 times longer than mine, and yet they have lifespans 40 times shorter than mine because they lose telomeres faster, change their pattern of gene expression faster than I do. In fact, you can accurately predict the lifespan of almost any species based on the rate of telomere loss. Telomere length is irrelevant. Relative telomere length turns out to be important only because it changes the pattern of gene expression. This is what looks like a very complicated diagram taken from my article. And really what it says is there are a number of upstream mechanisms and downstream biomarkers. Now the upstream mechanisms all play a role in the rate of aging. For example, abnormal microbiome, elevated blood glucose, hypertension, Trauma, for example, head trauma increases your risk of a number of dementias. Toxins, as I say, paraquat increases your risk of Parkinson's. Chemotherapy, genetic predispositions, infections, I could include not only the 4 listed there, but prions as well. Senolytic therapy tends to increase the risk actually of age-related changes. So radiation increases your skin aging. All of these play a role in aging. And downstream we have outcomes, for example, reactive oxygen species, a lower lipid turnover, tau tangles, beta amyloid. These are well-known biomarkers. Let me say something odd, which is none of this matters. To me, the question is not understanding the complexity of this, but the question is, where's the single most effective point of clinical intervention, both clinically and financially? And the answer is the upstream targets have proved inefficient. For example, if I entirely control your blood glucose and your blood pressure, if I prevent you from having any head trauma, if I make sure you don't get exposed to toxins, you still have a risk of, for example, Alzheimer's disease. No matter what we've tried upstream, even altering genes, we've never been able to show that this has a lasting significant impact on the total risk of, for example, age-related dementias. The same is true downstream. There have been effects trying to work with reactive oxygen species that haven't had much effect on the course of the disease. The same is true if we look at beta amyloid plaque. For example, there have been more than 1,100 interventional trials in Alzheimer's, none of which by global consensus have been able to demonstrate a significant impact on the course of the disease. So it's not that these are bad targets, they're just not efficient targets. The question is, where's the most efficient target? And the answer turns out to be age-related systems themselves, the aging process at a fundamental level. We know that both because it works in human cells and human tissues and in animals. This is the first example 22 years ago from the classic Bodnar paper, in which she showed that if you reset telomeres, you reset the pattern of expression of genes. You reset the epigenetic pattern. In fact, you take human cells and you make them young again. The same was true of tissues, first done 20 years ago this year by Walter Funk in his lab in California at Jaron, which you showed that human skin, old skin, can be turned into young skin if we reset telomere length and reset gene expression. The same is true of osteocytes. And finally, the same is true of vascular endothelial cells with a Matsushita article back 19 years ago. So we know we can reset aging in human cells. We know we can reset aging in human tissues. The question is, can we do it in animals? There are a number of interesting studies that have been done using telomerase activators over the past 4 years, peer-reviewed studies, 4 of them I can think of. The best one that came out that was useful was 2011 paper from Ron DePinto at Harvard, but he used a technique I can't use on human beings. Whereas a friend of mine, Maria Blasco, in this De Jesus article, Maria is the head of CNIO in Madrid, did this. And what she showed is that she could reset aging in mice and reset cognitive decline in mice effectively using telomerase in mice. So what we're going to do is the same thing. Let me point out this entire area is called Xenotherapeutics, not Xenolytics, where you actually just simply kill dead cells, but you take older cells and you resuscitate them. You effectively turn them into younger cells. It works very nicely. What we're going to do is exactly that. We have a scientific advisory board that's globally well known. For example, Zavin Khachaturian is the editor-in-chief of Alzheimer's Dementia. Suzanne Hendricks actually designed some of the major pharma trials for Alzheimer's, for example, the Biogen eSize study. Joe Araujo runs the biggest animal study group when you go to it. Kurt Whittemore did this study with Maria Blasco and so forth. The same is true of our clinical advisory board. Stephen Arnold, professor of neurology at Harvard, Lon Schneider at USC Keck, Russ Swerdlow, all of these people have run Alzheimer's studies over the years, are well known, and I chose them for being skeptical. These are people who see the value of the model and want to see it tested rigorously, but they are not true believers any more than I am. We intend to show that we can reverse Alzheimer's if we can and test the model. We have talked to the FDA about this. We're ready to go to them formally and take this to human trials within the next year or two. It's a single dose gene therapy using an adeno-associated virus, using a standard normal telomerase human gene. And what we're targeting is glial cells and neurons in the brain. So what we are talking about is not simply being able to slow or stop Alzheimer's, but to show that we can reverse the cognitive decline in human patients. We intend to take this to human trials and see if we're right. So thank you very much for listening. If you have any questions or comments, please feel free to get in touch with me. I look forward to talking to you. Thank you very much.
Thank you, Dr. Michael Fossel, for the talk.
Thank you, sir.
Thank you. So any questions for Dr. Michael Fossel?
I have other questions you have asked.
Please, ma'am.
Yes, sir. Can I ask you a question? Because I'm listening for long and I'm just, you are working so hard for decades together to find out the real cause of the aging. But my simple question is, where in every disease, there are so many of the risk factors. And some, they are visible, converted into the real disease. And in some, there are risk factors, but they are not become the real disease. And what are the things? Because in throughout the things I'm focusing, you are talking on the question mark on the process of the aging. So what restricts to convert the risk factor into the reality?
Yeah, this is exactly the point that I was making in that Alzheimer's conference and in the paper, which is that risk factors are not the disease and biomarkers downstream are not the disease.
Yes,
the overall
very
disease
true.
is the whole process. So for example, I know that, let me take a non-CNS example. I know that if I play basketball or some other high impact sport, I will have trauma to the knee and I accelerate my risk of osteoarthritis. So I begin to understand some of the risk factor, but the mechanism involved has to do with the loss of chondrocytes. The remaining chondrocytes divide, they age faster, they become more and more dysfunctional. You begin to get inflammatory changes. They're less apt to be able to make some of the proteins that are critical to joint function, and the outcome is disease. And we can essentially treat that disease by simply replacing your knee, but that doesn't treat the disease process. It simply removes it. It's like a heart transplant. That doesn't treat coronary artery disease. It just replaces the heart, or for that matter, coronary bypass grafts. So anytime I look at dementia, I find the same thing. There are a lot of upstream risk factors, and a classic example is head trauma. For example, American football players or hockey players, rugby players have an increased risk of this. And people tend to know that just the way they know their upstream genetic risk factors without being able to explain why. And my point is, if we understand the exact mechanisms of that, why is it that ApoE4, as I've just explained, increases your risk, for example, of beta amyloid microaggregates? Once we understand the process, the question becomes, where is the most effective point of intervention? And there are clinical trials going on right now that I was talking with someone about yesterday who's doing this, where they're trying to actually replace some of your APO4 alleles with APOE2 alleles. But that's like saying I'm going to prevent you from getting head trauma. It doesn't. It's probably not the optimal point of intervention. It's a point of intervention. For example, again, with vascular disease, if I can control your blood pressure and your blood glucose, I can lower your risk of a number of vascular diseases, but I don't prevent it. I just lower that risk or postpone it. You still may end up with myocardial infarction or stroke or aneurysm or vascular dementias. And so there's a worthwhile interventions, but I think that if we understand thoroughly the mechanisms, we can find more effective point of interventions. Again, for me, this is not an academic issue. It's a practical clinical one.
Okay, sir. That means I understood that you need to lower the risk by doing, understanding the mechanism and putting all the factors into to prevent that, then only you can prevent that disease and convert it into the reality.
Well, I think that, again, it's a question not just of lowering the risk, but of actually stopping it. So, with regard to the COVID question that came up in the last talk, we know that the older you are, the higher your risk of COVID, right? And there are a number of other risk factors that have been proposed over the past 12 months, some of which have been borne out, some of which haven't. But let's say that I could identify 3 major risk factors for you to prevent COVID. One of them would be having contact with other people. And clearly those are worthwhile risk factors to intervene in. But if I said, we can lower your risk factors versus giving you a COVID vaccine that was 100% effective, which we don't have, but if we did 100% effective with no side effects, which would you take? And the answer is the COVID vaccine. Once we understand the optimal point of intervention, that's the place to go for. In the meantime, where we are now with regard to things like Alzheimer's is trying to find ways of lowering risk factors and dealing with biomarkers. And that's a certain value to it, but it's like treating COVID by trying to avoid or lower your risk factors. It's not the optimal point of intervention. And at the same time, it's understandable.
Thank you very much, sir. Thank you for giving me the new insight. Let's see how will I do it tomorrow.
See what we can do.
Thank you, sir.
Thank you, Dr. Michael, for the talk, and thank you, Dr. Anjana Agarwal.
Thank you so much.
Thank you. So